Hello and welcome to the Talking Techniques podcast. Brought to you by Biotechniques, this show brings you the latest from the frontiers of the life sciences, straight from the people exploring them. I'm Beatrice Bolby, Biotechniques assistant editor and host of today's podcast. In this episode, supported by Sinobiological, we will be discussing CAR cell therapies.
CARs are synthetic receptors designed to redirect immune cells toward specific targets, typically cancer cells.
Their advantages and disadvantages.
By genetically engineering the T cells or the NK cells to express chimeric antigen receptors, these therapies can specifically recognize and bind to antigens present on cancer cells. While they have demonstrated remarkable success in certain types of blood cancers, CAR-T and CAR-NK therapies have shown limited effectiveness against solid tumors.
And their ethical considerations.
The process of selecting eligible patients raises questions of equity, bias, and transparency. Ensuring fair access to therapy, avoiding discrimination, and maintaining transparency in decision-making are crucial ethical considerations.
Our guest today is Ritvika Biswas, Field Product Scientist at Sinobiological. It's great to have you on the show.
Hi, Beatrice. Thank you for having me here today.
So just to start us off, can you please give us an introduction to CAR-T therapy and how it differs from other forms of immunotherapy?
Certainly. So first and foremost, CAR-T therapy is all about personalization. It starts by collecting a patient's own T cells, the soldiers of our immune system, and genetically modifying them in the lab. These modified T cells are then armed with chimeric antigen receptors, or CARs, which are designed to recognize specific antigens present on cancer cells. Now, this personalized approach ensures that the therapy is tailored to each individual, targeting their specific cancer. Now, unlike other immunotherapies that rely on general immune stimulation, CAR-T therapy takes a direct and precise approach. By equipping T cells with CARs, they gain the ability to specifically recognize and attack cancer cells, leaving healthy cells unharmed. Now, this targeted approach minimizes potential side effects and enhances the therapy's effectiveness. But there's more. So CAR-T therapy provides an extra boost to T cell activation. The CARs engineered into the T cells contain signaling domains derived from the T cell receptor, signaling components, along with co-stimulatory domains. This combination supercharges the T cells, ramping up their anti-tumor response and unleashing their full potential against cancer cells. Now, another standout feature of CAR-T therapy is the potential for long-lasting effects. These modified T cells can persist in the body, acting as vigilant guardians, continuously hunting down cancer cells as they appear. Now, this persistence and memory formation can contribute to prolonged remission and durable responses, setting CAR-T therapy apart from other treatments. Speaking of treatments, CAR-T therapy has shown remarkable success in certain types of blood cancers, like B-cell acute lymphoblastic leukemia and certain non-Hodgkin lymphomas. It has achieved impressive response rates and even complete remission in patients who have relapsed or failed other therapies. While CAR-T therapy holds great promise, it also presents unique challenges, managing potential toxicities, optimizing manufacturing processes, and expanding their application to solid tumors are areas of active research and development. It's a game-changing immunotherapy that harnesses the power of our immune system in a personalized, targeted, and potent manner. And very much like CAR-T is the CAR-NK therapy.
Yes, well, just going on to the next question then, what is CAR-NK therapy and how does it differ from CAR-T therapy?
So unlike CAR-T therapy, CAR-NK therapy utilizes a different type of immune cell called natural killer cell, or NK cell. Now, NK cells possess a remarkable ability to recognize and eliminate cancer cells without the need for prior activation or specific antigen recognition. In CAR-NK therapy, NK cells are obtained from a donor and undergo genetic modifications to express CARs that specifically target cancer cells. So once these genetically modified cells are introduced into the patient's body, they engage in a vigorous battle against the cancer cells. What sets CAR-NK therapy apart is its potential for fewer side effects compared to CAR-T therapy. This is because NK cells are less likely to induce a potentially dangerous immune response known as the cytokine release syndrome, or CRS. Another advantage of CAR-NK therapy is its potential for utilizing NK cells from a variety of donors. This widens the availability of this therapy as it's not restricted to using the patient's own T cells like the CAR-T therapy. However, it's essential to note that CAR-T therapy has undergone more extensive clinical trials and has already gained approval for treating certain types of blood cancers from the FDA. On the other hand, CAR-NK therapy is still in the early stages of development. As ongoing research progresses, it holds tremendous promise as a safe and effective treatment option for a wide range of cancers. So there you have it. CAR-NK therapy and CAR-T therapy may share a common goal of genetically modified cells to treat cancer, but their utilization of different immune cells and the respective stages of development distinguish them in the realm of immunotherapy.
Excellent. So just to take a few steps back then, what actually is CAR and how is it developed in these cell therapies?
Okay, so CARs, for chimeric antigen receptors, are synthetic receptors designed to redirect immune cells toward specific targets, typically cancer cells. The development of CARs involves several important steps that contribute to their effectiveness in killing cancer cells. Firstly, the process begins with target selection. Researchers identify suitable target antigens present on the surface of cancer cells. These antigens should ideally be specific to cancer cells while avoiding normal healthy cells. Now, commonly targeted antigens include CD19, found in B-cell malignancies, and HER2 overexpressed in certain solid tumors. Once the target antigen is chosen, the CAR is designed. A typical CAR comprises several key components. The extracellular targeting domain, which is responsible for recognizing and binding the target antigen, often derived from an antibody with a specific affinity for the target. The spacer and the transmembrane domains provide structural support to the CAR, connecting the extracellular domain to the intracellular signaling domain. Now, the intracellular signaling domain is a crucial component that triggers cellular activation upon antigen recognition. The commonly used signaling domain is derived from the CD3 zeta chain of the T-cell receptor complex. An additional co-stimulatory domain such as CD28 or 41BB may also be incorporated to enhance CAR-T or CAR-NK cell functionality. After the CAR design phase, the next step is CAR delivery into T cells or NK cells. This is achieved using gene transfer techniques, with viral transduction being the most common method that's used. In viral transduction, a viral vector, often a lentivirus or retrovirus, carries the genetic information encoding the CAR into the immune cells. Now, this viral vector infects the cells and integrates the CAR construct into their genome. Once the CAR is successfully introduced into the immune cells, they undergo ex vivo expansion in the laboratory. This step is crucial to generate a large population of CAR-expressing cells for therapy. Through careful cultivation and stimulation, the modified T cells or NK cells are grown and expanded to achieve the desired cell numbers. Finally, the CAR-T or CAR-NK cells are ready for infusion back into the patient. Upon encountering cancer cells expressing the target antigen, the CAR on the immune cells recognizes the antigen and triggers a response, leading to the destruction of the cancer cells. It's important to note that CAR development is an ongoing process, with researchers continuously refining and improving CAR technologies. Variations in CAR design and delivery techniques may exist based on specific research or clinical trials. As scientific advancements continue, we anticipate even more exciting developments in the field of cellular therapies.
So what are the advantages of CAR-T and CAR-NK cell therapies?
CAR-T and CAR-NK therapies offer a multitude of benefits in the realm of cancer treatment. So let's just talk about some of these advantages. Firstly, both CAR-T and CAR-NK therapies exhibit precision targeting. By genetically engineering the T cells or the NK cells to express chimeric antigen receptors, these therapies can specifically recognize and bind to antigens present on cancer cells. This targeted approach enables the destruction of cancer cells while minimizing harm to healthy cells. Secondly, CAR-T and CAR-NK therapies exert potent anti-tumor activity. Upon encountering cancer cells expressing the targeted antigens, these modified immune cells initiate a robust immune response. They release cytotoxic molecules and recruit other immune cells, intensifying the attack on the tumor and enhancing the overall anti-tumor effect. Now, another notable advantage is the persistence and long-term activity of CAR-T and CAR-NK cells. These cells can remain active within the body for an extended duration, providing ongoing surveillance against the cancer cells. So this persistence enhances the potential for durable responses and long-term remission in patients. CAR-T and CAR-NK therapies have also demonstrated promise in overcoming treatment resistance. By targeting specific antigens, they bypass mechanisms that cancer cells employ to evade traditional treatments like chemotherapy or targeted therapy. This ability to overcome resistance opens new avenues for effective cancer treatment. Furthermore, CAR-T and CAR-NK therapies exhibit broad applicability across various cancer types. They have shown efficacy in targeting hematological malignancies such as leukemia and lymphoma, as well as solid tumors like breast, lung, and ovarian cancers. This versatility makes them valuable treatment options for a wide range of patients. Moreover, CAR-T and CAR-NK therapies offer adaptability and potential for combination approaches. Researchers can engineer CARs to express different co-stimulatory molecules and cytokines, tailoring the therapies to optimize their effectiveness. Additionally, these therapies can be explored in combination with other treatments, such as checkpoint inhibitors or immune modulating agents, or to further enhance their impact. While CAR-T and CAR-NK therapies bring remarkable advantages, it's important to acknowledge the challenges associated with their development. Managing potential toxicities and refining manufacturing processes are ongoing areas of focus in research and clinical trials. By addressing these challenges, researchers strive to improve the efficacy and safety of these innovative therapies, opening new horizons in the battle against cancer.
Brilliant. Well, now, of course, we have to talk about their disadvantages.
Oh, yeah, sure. Let's discuss some of the potential limitations and challenges. Firstly, one of the major concerns is the potential for toxicity. CAR-T cells can trigger a strong immune response, leading to a condition known as CRS and neurotoxicity, like I talked before. These side effects can range from mild to severe, and in rare cases can be life-threatening. It's crucial for healthcare professionals to closely monitor and manage these potential toxicities during treatment. Another disadvantage is that CAR-T and CAR-NK therapies have shown limited effectiveness against solid tumors compared to blood cancers like leukemia and lymphoma. While they have demonstrated remarkable success in certain types of blood cancers, the response rates for solid tumors have been relatively low. Further research and advancements are needed to enhance their efficacy in treating solid tumors, and we'll discuss this later. Cost is another consideration. CAR-T therapy in particular is a complex and expensive treatment. The high cost may limit accessibility to all patients, posing a challenge in ensuring widespread availability and affordability. Addressing the cost factor is crucial and ensure equitable access to these innovative therapies. Additionally, the long-term side effects of CAR-T therapy are not yet fully understood. This is a new therapy. It's important to continue studying the impact of CAR-T therapy on the immune system and other organs over an extended period. Monitoring and managing potential long-term side effects are vital aspects of patient care. Then limited availability is also a challenge. The production of CAR-NK cells is still a very complex and costly procedure. Furthermore, CAR-T therapy is a specialized treatment that requires specialized facilities and expertise, which may limit its availability to all patients. Efforts are being made to improve manufacturing processes and expand access to these therapies. There is also a concern of antigen escape, where some cancer cells can downregulate or alter the target antigen recognized by CAR T cells, potentially leading to cancer relapse. Overcoming this challenge is an effective area of research to ensure sustained effectiveness of CAR-based therapies. Lastly, for CAR-NK therapy, further optimization is needed. The design and function of CAR-NK cells are still being studied and refined. Researchers are working to determine the most effective approaches in terms of CAR design, target selection, and manufacturing processes. So basically, that was an extensive list of challenges that researchers are currently striving to address. And in the healthcare industry, it is a shared objective to not only pioneer effective and innovative therapies, but also ensure that these are accessible to all individuals. As a dedicated biotech company, we at Sinobiological are committed to contributing to this collective effort by supplying researchers with exceptionally high-quality GMP-grade cytokines. And these cytokines play a vital role in the development of CAR in CAR T and CAR-NK therapies. What sets us apart is our unwavering commitment to offering these critical components at highly affordable prices. By doing so, we actively contribute to reducing the overall cost of these therapies.
And how do you think we can improve the safety and efficacy of these cell therapies?
So that's another area of focus, which includes the development of new CAR designs, such as dual targeting CARs, which can recognize multiple targets on cancer cells, and the use of gene editing technologies to improve the function and persistence of CAR T cells. Maybe I should elaborate on that a little bit. CAR T therapy has shown promising results in treating certain types of cancers, but there are still challenges to overcome in terms of safety and efficacy. This is something that we did not really talk about when we were discussing the limitations of these cell therapies, which is that sometimes CAR T and CAR-NK cells target healthy cells along with cancer cells, leading to side effects. Dual targeting CARs, which can recognize multiple targets on cancer cells, may help reduce off-target effects and improve the specificity of CAR T therapy. In addition, the use of gene editing technologies such as CRISPR-Cas9 can help improve the function and persistence of CAR T cells. For example, researchers are exploring ways to use gene editing to delete certain genes in CAR T cells that may cause them to become exhausted or to target healthy cells. Gene editing can also be used to introduce modifications to CAR T cells that enhance their anti-tumor activity or to make them resistant to the immunosuppressive environment of tumors. Overall, these advancements in CAR T therapy are exciting and hold great potential for improving the treatment of cancer. However, it is important to continue to carefully evaluate the safety and efficacy of these new therapies in clinical trials before they become widely available.
And what are the ethical considerations then around CAR T and CAR-NK therapies?
So since these cell therapies involve genetically modifying a patient's own T cells and NK cells to recognize and attack cancer cells, the use of CAR T and CAR-NK therapy raises several ethical considerations that arise in its use, particularly in relation to patient selection and access to treatment. Just for the ease of understanding, let me talk only about CAR T therapy here. Firstly, patient selection is an important ethical concern. CAR T therapy is currently approved for specific types of cancer and is typically used as a last resort for patients who have not responded to other treatments. The process of selecting eligible patients raises questions of equity, bias, and transparency. Ensuring fair access to therapy, avoiding discrimination, and maintaining transparency in decision-making are crucial ethical considerations. Access to treatment is another significant ethical consideration. CAR T therapy is a complex and expensive procedure, which can create barriers to accessibility. The high cost of therapy may result in disparities, limiting access based on socioeconomic status. Ethical imperatives include ensuring equal access to CAR T therapy for all eligible patients, regardless of their financial resources. Addressing affordability issues and exploring measures to reduce the financial burden are vital ethical concerns at the moment. Informed consent is a fundamental ethical principle in CAR-T therapy, so that's another thing. The treatment involves substantial risks and potential side effects, including life-threatening complications in some cases. Patients and their families must be fully informed about the potential benefits, risks, uncertainties, and alternative treatment options. Ethical considerations include prioritizing clear communication, comprehensive patient education, and providing adequate support to facilitate well-informed decision-making. Long-term follow-up and data collection are important ethical considerations in CAR-T therapy as well. As a relatively new treatment modality, the long-term effects and durability of response are still being studied. Ensuring appropriate long-term monitoring and data collection is essential to improve therapy and inform future patient selection. Ethical responsibilities include promoting data transparency, sharing research findings, and continuously evaluating and refining the therapy based on emerging evidence. Now, global disparities in access to CAR-T therapy pose ethical challenges as well. Currently, the therapy is more accessible in developed countries with advanced healthcare systems. Bridging the gap between developed and developing nations in terms of technology transfer, infrastructure development, affordability, and regulatory frameworks is crucial to ensure a more universal and equitable availability of CAR-T therapy. And these ethical considerations underscore the importance of careful deliberation, transparent decision-making processes, equitable access, and ongoing evaluation of CAR-T therapy. By addressing these concerns, we can maximize the benefits of CAR-T therapy while navigating the potential ethical challenges it presents.
And are there any other applications of CAR-T therapy besides cancer?
Actually, yes. So CAR-T therapy has demonstrated remarkable success in cancer treatment, but its unique mechanism of action can be harnessed for other diseases as well. One area of exploration is CAR-T therapy for autoimmune diseases. By engineering CAR-T cells to target and eliminate specific immune cells involved in autoimmune reactions, researchers aim to modulate the immune response and restore balance in conditions like rheumatoid arthritis, multiple sclerosis, and type 1 diabetes. Preliminary studies have shown promising results, although more research is needed to optimize the approach and ensure safety. Another exciting application of CAR-T therapy is in the field of infectious diseases. By equipping CAR-T cells with receptors that recognize viral or bacterial antigens, scientists aim to enhance the immune response against these pathogens. This approach could potentially provide a novel and targeted therapy for viral infections such as HIV, hepatitis B, cytomegalovirus, as well as bacterial infections that are challenging to treat with traditional antibiotics. Additionally, CAR-T therapy is being explored in the context of organ transplantation. Transplant rejection occurs when the immune system recognizes the transplanted organ as foreign and mounts an immune response. By engineering the CAR-T cells to specifically target and eliminate immune cells involved in rejection, researchers aim to prevent or mitigate transplant rejection and improve the long-term success of organ transplantations. Furthermore, CAR-T therapy holds promise in treating certain hematological disorders such as hemophilia and sickle cell disease. By modifying CAR-T cells to produce and deliver therapeutic proteins or correct genetic mutations, researchers aim to address the underlying causes of these conditions and provide long-term benefits for patients. Now, while these applications are still in early stages of development, they highlight the versatility of CAR-T therapy and its potential to revolutionize treatment approaches beyond cancer. Continued research and clinical trials will be instrumental in advancing these novel applications and bringing them closer to clinical reality.
Well, that's excellent. Thank you. So can you give us a little bit of a summary of what we've talked about today and then what you think the future holds for CAR cell therapies?
Sure. So CAR T therapy is a revolutionary approach to cancer treatment that involves genetically modifying a patient's T cell to attack cancer cells. While CAR T therapy is currently only approved for the treatment of certain types of blood cancer, there is a lot of excitement about the potential of this therapy to treat other types of cancer and even other diseases beyond cancer. One potential application of CAR T therapy in the future could be the treatment of solid tumors, such as those found in lung, breast, and colon cancer. They are more difficult to treat with CAR T therapy because they have a different tumor microenvironment than the blood cancers. However, researchers are exploring ways to modify CAR T cells to better target solid tumors and to overcome the challenges of the solid tumor microenvironment. Another potential application of CAR T therapy is in the treatment of autoimmune diseases, like we talked about. In the near future, CAR T therapy could offer a targeted and potentially curative treatment option for rheumatoid arthritis, type 1 diabetes, etc. Then advancements in CAR T technology are also being made to improve the safety and efficacy of the therapy. For example, researchers are exploring ways to engineer CAR T cells to be less toxic and to better target cancer cells. They are also investigating ways to improve the manufacturing processes to make CAR T therapy more accessible and affordable for patients. Overall, the future of CAR T therapy is really bright, and there is a lot of excitement about its potential to revolutionize the treatment of cancer and other diseases. As research and development continue, we can expect to see further advancements in CAR T technology and more potential applications of this therapy in the coming years.
Well, Ritvika, it's been great speaking with you today and learning more about CAR cell therapies. Thank you so much for coming on the podcast.
Thank you so much for having me here.
If you have enjoyed this episode and would like to find out more about CAR T and CAR NK cell therapies and their development, you can check out our In Focus with Sinobiological at www.biotechniques.com. Thank you for listening and goodbye.